Good afternoon, dear colleagues. Welcome to our monthly dialogue. Dialogues have been a successful feature of the European Heart Journal. You know, as all editors, since the beginning, we believed that the mission of the European Heart Journal is not just publishing top science, which is extremely important, but is not enough. We also tried another mission we developed has been to create a link between editors, authors, reviewers, and colleagues and cardiologists and readers. A link which helps in implementing our mission, which is to reduce the burden of cardiovascular disease. Therefore, not just top science, but also dialogues. Dialogues are monthly webinars typically with the leaders in cardiology from the 5 continents. And in these dialogues, our goal is not only to unveil the science produced by our guests, but also their human side, because their life experience, the way they developed their scientific project, the interest in cardiovascular medicine, the human side of the story, we believe is very inspirational, in particular for our younger colleagues. Now, today we have the monthly dialogue. I remind you, it's always on the last Thursday of each month at 5 o'clock Central European Time. And today we have, as usual, a very important guest. The guest this month is Professor Carlo Patrono. I will host him with Lina Badimon, who is Executive Editor of the European Heart Journal. And today we have 2 protagonists. One is Carlo Patrono and the other is aspirin. The title of this dialogue is The Long Journey of Aspirin in Cardiovascular Disease, narrated by Professor Carlo Patrono, the protagonist in this long history of aspirin. Now, Lina, please, can you introduce Carlo?
With great pleasure. I mean, Carlo has been a role model for me since I was very young. So I met him in the United States, and it was really love at first sight, I have to say, on his science and his person as a gentleman. So for me, it's a great pleasure to have this dialogue with him. So Professor Carlo Patrono received his MD degree from the Catholic University School of Medicine in Rome, in Italy, and trained as a postdoctoral fellow at Mount Sinai School of Medicine in New York with the late Solomon Berson and Nobel laureate Rosalind Yalow. He was visiting professor at the Royal Postgraduate Medical School in London afterwards, also at Mount Sinai in New York, and at the University of Pennsylvania in Philadelphia. He has been going there continuously during all his scientific life. So he's presently adjunct professor of cardiovascular pharmacology at the Catholic University in Rome. His main research interest is the study of platelet activation and initiation in atherothrombosis and the role of platelets as well in colorectal cancer. His research has characterized the human pharmacology of aspirin as an inhibitor of platelet thromboxane production and provides the basis for the development of low-dose aspirin as an antithrombotic agent. So his job was really important for cardiology. Moreover, Professor Patrono contributed to the characterization of the human pharmacology of selective cyclooxygenase 2 inhibitors and contributed to the evaluation of their effects in different clinical settings. He is currently working on the hypothesis that platelet inhibition may represent a key mechanism of action underlying chemoprevention of colorectal cancer by low-dose aspirin. He is an elected member of the Association of American Physicians, the Royal College of Physicians, the Accademia Europea and Accademia Nazionale dei Lincei. He shared with Dr. Garrett Fitzgerald a long-standing friend of Carlo, the 2013 Grand Prix Scientifique awarded by the Institute of France for his work on low-dose aspirin. So internationally recognized for his work. He's a deputy editor of the European Heart Journal and section editor of the Journal of the American College of Cardiology. Professor Patrono has published over 250 research documents with over 98,000 citations and has an age index of 118. So his trajectory has been a very successful one, and I think we are really very, very happy to be able to have this dialogue with him today. So I think I'm going to start with questions regarding this trajectory in life. So I wouldn't say the first love, but close to that, was prostaglandins. So he liked prostaglandins very much. So one of your first publications appeared in 1973 and was on the one immunoassay to measure prostaglandin F2. So you have continued with this interest in prostaglandins for the majority of your life. So what triggered your interest on prostaglandins, Carlo?
Thank you for your kind introduction, Lina. So Angela, the love of my life, came before prostaglandins, but shortly after, just a few years later, when I graduated in medicine and decided to do an academic career in pharmacology, I had an opportunity of attending a international pharmacology meeting in Florence. And there I heard for the first time John Vane and Bengt Samuelsson, a pharmacologist from London and a medicinal chemist from Stockholm, talk about their prostaglandin work. I was fascinated by this research on prostaglandins. The only prostaglandins known at the time were prostaglandin E2 and prostaglandin F2 alpha. So it was just the surface of the prostaglandin family. And yet it was intriguing that this was apparently an ubiquitous regulatory system. In physiology, for example, there was evidence that prostaglandin E2 participated in gastrointestinal cytoprotection, as well as in pathophysiology, there was evidence that the same prostaglandin was involved in pain and inflammation. So that sounded very interesting. And I said, maybe this is something to keep an eye on, which I did eventually.
Well, indeed, it was a fascinating time because all of the other prostaglandins had to be discovered. And then the Nobel Prize went to Samuelsson and Vane in 1982. This was 9 years after your first paper on prostaglandins. And the story has been successful so far because then all of them came, they were, as you say very well, regulators of different functions, and then you kept interested. And I know with your team, you have been providing a lot of information on how these prostaglandins work. So in your view, what has been your major contribution to our knowledge of prostaglandin, your own work?
Well, based on my experience with Journal Dialogue that you briefly mentioned, and my exposure to their fantastic analytical method, the radioimmunoassay, I very early started developing antibodies against different prostanoids. And when Bengt Samuelsson discovered thromboxane A2 as the major metabolite of arachidonic acid in human platelets, I became interested in trying to measure thromboxane biosynthesis in humans. Thromboxane A2 as such is chemically unstable. It disappears with a half-life of 30 seconds. And it's highly potent, so there may be a reason for this high chemical instability. And so we developed antibodies against its chemically stable, high-duration product, thromboxane B2. And we developed further analytical methods to measure its major urinary metabolites. So one of the important contributions my lab made to the prostaglandin field was characterizing thromboxane biosynthesis and inhibition in humans, both in health and disease, particularly cardiovascular and cerebrovascular disease. We did that by two different routes. One was developing a ex vivo biomarker to assess the pharmacology of aspirin, that is measurement of serum thromboxane B2. We made this very naive and simple observation that thromboxane B2 is not present in plasma. These are not circulating substances, but locally active autocoids. But if you let whole blood clot at 37 degrees for 1 hour, you get an explosion of platelet thromboxane production, and you measure hundreds of nanograms of thromboxane B2 in serum. And so this gives you a very high signal that can be completely abolished by administration of aspirin. So an ideal biomarker to assess the clinical pharmacology of aspirin. Then we did infusion studies onto ourselves by infusing this biologically inactive derivative thromboxane B2 to characterize its fate in the human circulation, its rate of disappearance, its enzymatic metabolism by the liver, and characterize a few enzymatic metabolites which are excreted in the urine and that can serve as an index of thromboxane biosynthesis in vivo. And what we further showed is that these metabolites can reflect in vivo platelet activation. And this has had profound impact in the field.
Yeah, that has affected the work of many others that follow your path. So meeting Rosalind Yalow was an experience, right? Because when I went to Mount Sinai, everybody talked about her, but she wasn't there any longer. So I didn't have the opportunity of meeting her. How was she? Because she really was great in the application of the enzyme immunoassays and all these biochemical evaluation. So how was she as a person?
Well, she was a fantastic woman, capable of integrating into her daily life a passionate interest in research, with continuing to care for her family. He had a husband and children waiting for her to make dinner at night, but then she would come back to the lab at 8 o'clock in the evening and continue working. And there was the time when Saul Burson, who had become chair of medicine at Mount Sinai, used to join her to run joint radioimmunoassays for the very early hormones that they characterized, initially insulin, then ACTH, parathyroid hormone, growth hormone, and so on and so forth.
Yes.
And the very first year I was in their lab, I was living in the hospital, which was the Bronx VA hospital affiliated with Mount Sinai, and where the nuclear medicine lab was located. And really, the integration of the expertise of Rose Yalow, who was a physicist expert in nuclear physics, with Saul Burson, an MD endocrinologist, who was interested in studying insulin metabolism in diabetes, was the key to their success in developing this unique method that combines the sensitivity provided by the use of radioactive substances with the specificity provided by specific antibodies.
Yeah. So now it's not so used, right? Because of the radionuclides is not, but she opened the path
Yes.
to others, right?
And the antibody part of it has remained.
Exactly.
And,
So,
and you know, what she taught me was not only this fantastic analytical method to measure virtually any substance you may think of in human biological fluids, but also the importance of focus and persistence.
yeah, well, that's good for our audience. focus on persistence is very good. Yes. Okay. So she was the second woman that got the Nobel Prize. So it was very good for you to have been able to meet her. So now maybe Filippo, you want also to have some dialogue with Carlo.
Thank you, Lina. Well, Carlo, when I read the word aspirin, I think of you. Must be some sort of Pavlovian conditioned reflex in my mind. Neurons linked, have been reshaped. When I read aspirin, I think of you. Now, your first paper on aspirin was in 1975. How did you fall in love with aspirin?
Well,
and why?
I told you my fascination with the early work of Bain and Samuelson. Then came 1971. I was still in New York in the lab of Berson and Yao, and 3 papers appear in the same issue of Nature describing, for the first time, molecular understanding of the mechanism of action of aspirin. This was the work of John Vane and his associates in London, describing the fact that aspirin and other aspirin-like drugs like indomethacin or ibuprofen at therapeutic concentrations inhibited prostaglandin synthesis in vitro in various cellular and tissue systems. And they proposed, John Vane proposed in particular, that this would explain the analgesic, antipyretic, anti-inflammatory effect of aspirin, as well as its potential gastrointestinal toxicity because of the physiological cytoprotective role of prostaglandins I briefly mentioned in the beginning. So that was a turning point in my interest on aspirin. And after Banks Samuelson discovered thromboxane in human platelets in 1975, I became interested in comparing different tissues for the sensitivity to aspirin, because there was already evidence that the different pharmacological effects of aspirin required markedly different doses over a range of about 100-fold, from the very low concentrations required for an antiplatelet effect to the very high doses required for an anti-inflammatory effect. And I started playing with isolated platelets from circulating blood and looking at fragments of synovial tissue obtained from our colleagues in orthopedic surgery at the Catholic University. And we did find and published about 1975-76 a paper describing differential inhibition of prostaglandin synthesis in platelets versus synovial tissue. And then we rapidly moved into the in vivo situation by developing the methods I just briefly discussed with Lina, which enabled us to explore the clinical pharmacology of thromboxane inhibition in healthy subjects and then in patients with cardiovascular disease. So I would say it was love at first sight in 1971 when John Vane made the discovery of the mechanism of action, and then another episode of love at first sight when Banks Samuelson discovered thromboxane A2 as a major prostanoid made by human platelets, and therefore established for the first time a link between aspirin inhibiting prostanoid production and inhibiting platelet function, because none of the prostaglandins known until that time had any effect on platelet aggregation, but thromboxane did. And thromboxane was a very powerful platelet aggregating substance.
Carlo, when you propose something really new, when you want to change a paradigm in the history of medicine, cardiovascular medicine, the initial reaction of the scientific community is critical. The new thing, in particular, if it is important, generates a sort of defensive reaction, which tends to preserve the current paradigm. Now, how difficult was in your case to convince the scientific community that low-dose aspirin would have changed the history of cardiovascular prevention? Was it an easy game or a difficult game?
It was a very difficult game. And you know, I went to meetings, and the typical reaction I would get was, oh, these are interesting biochemical findings, but have nothing to do with what happens in the patient. And the popular theory at the time, and I'm talking about late 70s, early 80s, was that aspirin had many pharmacological effects that were dose-dependent, that required higher doses given several times a day. And this was the key to its potential antithrombotic effect. And in fact, the very first randomized clinical trials of aspirin were done by people in the US and in Canada using doses as large as 1,000 to 1,500 milligram daily. This was the case of the early trials in cerebrovascular disease, and the champion there in Canada was Harry Barnett. And the first trials in patients undergoing coronary bypass surgery, the early trials of Valentin Fuster and Jim Chisborough. And their choice of these high doses of aspirin was driven by some basic scientists in Canada who published data suggesting that there were these extra platelet effects of aspirin that really played an important role. And the company marketing aspirin went behind this trend. And so even the company making aspirin was against the story that, look, this is completely wrong. The way to go is by much lower doses, which were 15 to 30 times lower than the doses used in these early trials. I'm sure you will remember the AMIS trial in over 4,000 patients with a myocardial infarction. It was a mortality trial with 1 gram of aspirin daily. And mortality was not reduced, but in fact was numerically increased by high-dose aspirin. And that was 1980, and it sounded as the de profundis of aspirin in cardiovascular medicine. And it was difficult to resurrect it and convince the medical scientific community that there was perhaps a different avenue that one could walk safely.
Well, really a fascinating story. Well, when aspirin, just a curiosity, when aspirin became popular and accepted, do you remember if there was an increase in the cost of aspirin? Because this is happening now with colchicine, another drug which has been repurposed, as we know very well, and this is leading to a marked increase in the cost. What about aspirin? I don't remember now.
Well, there was some increase when the company decided to switch from plain aspirin to enteric-coated aspirin. And being a new formulation, it did command a new price, which of course was higher than the previous one.
But overall, the cost has remained much lower than many other drugs useful in prevention. But I initially said, well, in these dialogues, we want to unveil not only the science of our guests, but also their human side, because I believe that this is inspirational for our younger colleagues. And so let's come back to your personal life. I would say to our personal lives, because the first time we met, not in Rome at the Catholic University, but the first time we met at Hammersmith Hospital in London. invited by my mentor, Attilio Maseri. Tell me about Attilio. Did Attilio Maseri influence your career? What did you learn from Attilio?
Okay, before I talk about Attilio, let me briefly explain why I went there. Going back to Burson and Yellow, I just want to say a few words about Saul Burson, who unfortunately died prematurely before the Nobel Prize was awarded. At the end of the first year that I was there, and I had worked on a glucagon radioimmunoassay, I became so interested in prostaglandins that I told him, why don't we develop antibodies against prostaglandin E2 or F2 alpha? And he looked at me and said, you know, that's an interesting idea, and we will learn a new field. And this, we will learn a new field, has been an important lesson for me, who, as a clinical pharmacologist, have jumped from endocrinology at first to rheumatology because of nonsteroidal anti-inflammatory drugs, then to cardiology because of platelets, and more recently to oncology because of an apparent chemopreventive effect of aspirin. So when I decided I wanted to explore the effect of aspirin, low-dose aspirin in cardiovascular prevention, I told myself that I needed to learn a new field. And where else could I go to learn the new field of cardiology other than the cardiovascular unit of the Hammersmith Hospital, the Royal Postgraduate Medical School in London, headed by this wonderful man, Attilio Maseri. Attilio was really an inspiring example of a modern physician-scientist. And what I learned from him, besides understanding some of the pathophysiology of acute coronary syndromes and his work on alterations in coronary motility and reactivity to different stimuli, was never think in a box, because he was clearly a man who did not enjoy being in the mainstream, but rather opening new avenues. And then the interest in evaluating the individual patient, trying to understand pathophysiology in the individual patient rather than putting everybody together in the big randomized clinical trials. And being a pharmacologist, I translated that into trying to understand the determinants of the inter-individual variability in drug response. And so he had many influences on my further academic career when I returned to Rome, initially to Chieti and then back to Rome.
Well, this is the charm of academia. Mentors who teach trainees, then trainees become mentors of new trainees, and the history continues. I think this is fascinating. And this progression, continuous progression from mentors to trainees is really the salt of academic medicine and the origin of the incredible advancements we have done in the last 40, 50 years or more. This transmission of knowledge from mentors to trainees is really the quintessential truth of academic medicine, one of the reasons why we are progressing so well in the treatment of our patients. Lina, back to you.
Yeah, but also the point is that you never should stop learning. Yeah. It's true. I mean, every day you can learn. And this is what Attilio showed me, because I met him when he was old, but he impressed me because he was saying these things. I mean, well, this is already known. I mean, we have explained to me, what is this thing going on? What is, he was really an amazing person indeed. But going back to the idea of the dose, right, of aspirin, I mean, I believe that many drugs have been failing because the pharmacology has not been adjusted, you know? This is the idea that I have for many different drugs that we have developed in the past, and have been put out of the way because the pharmacology of how they have to be used is forgotten, you know? And I think this is something that we should go back and think about how to improve the use of some drugs, yeah.
If I can make a comment, Lina, on this very important point, I want to emphasize that the clinical development of low-dose aspirin was not driven by industry, but was driven by us, by the medical scientific community. And the main difference is that we carefully selected the right dose to test in clinical trials with a very extensive phase 2 that lasted about 8 years. Now, this would not be acceptable by industry, but we have to realize that many trials that fail in phase 3 are due to inadequate phase 2 studies. At least that's my own opinion.
Okay. We agree that aspirin had a dominant role in cardiovascular prevention, but now the aspirin use is challenged by newer antiplatelet drugs, like ticagrelor, prasugrel. Well, you know very well those new drugs. Now, my point is, what do you see? How the future is going to evolve? Is aspirin going to be withdrawn in the next few decades, or aspirin will remain as a coadjuvant?
You know, if I take a regulatory crystal ball, I would say that aspirin is here to stay because no other antiplatelet agent has been shown convincingly to be superior to aspirin.
Yeah.
Despite the fact that the industry, several companies have perform superiority trials versus aspirin, but none of these superiority trials, largely P2Y12 blockers, have shown superiority that was approved by a regulatory authority like EMA or FDA. So unless a new antiplatelet agent, which is not on the horizon, comes up over the next 10 years, that has a completely different mechanistic strategy to inhibit platelet function. And I would love to see such a development. I haven't seen it so far, but let's say that this may happen. If the company is willing to spend the money necessary to do a 20,000 patient trial to show superiority versus aspirin, this may displace aspirin from its preeminent role. However, if I take not a regulatory crystal ball, but a promotional crystal ball that is influenced by marketing pressures, by publication pressure, then I fear that low-dose aspirin may be eventually displaced from its current role because of fashionable trends. This reminds me of what happened in the 70s when the conventional aspirin we know when we buy in the pharmacy when we have headache or fever, that kind of aspirin was displaced by the analgesic market, by the newer non-steroidal anti-inflammatory drugs that are simply aspirin-like drugs, working by a similar mechanism of action. And they displace aspirin because of increased marketing pressure. So something similar may happen in the case of aspirin. And I would not be surprised if progressively people would say, well, aspirin is 125 years old. Maybe it's time for retirement, and we use a newer agent.
Okay, now another area is the COX-2 inhibitors. You work also with COX-2, and what is your opinion or your thought about the relevance, the therapeutic relevance of these drugs, the COX-2 inhibitors, in the different areas? Because in different areas they have a different relevance, right?
Well, the COXIBs, there is a new class of NSAIDs that were developed by several companies as selective COX-2 inhibitors, represent the single NSAID class that was most thoroughly investigated. I mean, typically a traditional NSAID was studied in randomized clinical trials over a few hundred patients. The COXIBs were studied in clinical trials of 10,000, 20,000, 30,000 patients. And what we know today about traditional NSAIDs is due to the COXIB trials, because typically COXIB would be compared to a traditional NSAID like ibuprofen or diclofenac or naproxen. And for example, the fact that these drugs can have untoward cardiovascular effects that may double the risk of a myocardial infarction is due to the COXIB trials. Because of their size and duration. There was a development in the COXIB area of exploring their potential chemopreventive effect against colorectal adenoma recurrence. So they did placebo-controlled trials of 3 years duration, and that's when a clear cardiovascular effect was seen. With rofecoxib, which led to the withdrawal of rofecoxib from the market. So they remain the most thoroughly investigated class of NSAIDs. They are the only ones with a proven improved gastrointestinal safety profile. But because of this potential cardiovascular toxicity, we cannot use them long-term. We cannot use higher doses of them.
Right. We have a question here from Dr. Gosa Tabaei. I'm sorry, I didn't pronounce your last name very well. So she's asking, do you want to speak? Yeah?
Yes.
Yeah, go ahead. Yeah, pose your question for Dr. Patrono.
Good evening. I wanted to ask, could aspirin be a good drug for a group of HFpEF patients with high ESR or blood viscosity? Because could aspirin be a good low ESR lowering drug? With your clinical insight, according to your
if I understand correctly, you're asking whether aspirin could be used in patients with heart failure with preserved ejection fraction, right?
Yes.
Well, it depends on the coexisting conditions of the patient that may or may not require having aspirin on board. I mean, I would hesitate to use aspirin in any clinical setting unless there are adequate randomized clinical trials to support its use. Because as you know, in addition to its beneficial effects, aspirin may have untoward effects intrinsically linked to its mechanism of action of inhibiting platelet function, which will increase the risk of bleeding from pre-existing lesions. In other words, aspirin will not cause a new bleeding if you don't have a lesion anywhere. But if there is any lesion anywhere, some lesion somewhere, for example, in the upper gastrointestinal tract, then aspirin will double the risk of bleeding from that lesion. And the same is true for other antiplatelet agents. So your idea is interesting. It could be tested. There were some trials in the field of heart failure, and the key person in this area is John Cleland. with some controversial findings.
Okay, so my last question to you, Carlo. Also, I want to see your personal view in addition of the scientific merit, right? So you share with Garrett Fitzgerald the 2013 Grand Prix, Scientific Prix of the Institut de France. So this was for your work on low-dose aspirin. So tell me, what was this, what the prize meant to you? Because this is a very important prize, right? It's close to the Nobel Prize. I mean, people that get this get the Nobel Prize. So what was your feeling? How did you experience that honor?
Well, the wonderful feeling was due to the fact that both my work and Garrett's work were recognized jointly, because at about the same time that we were doing the studies I described to you in Rome, Garrett was doing similar clinical pharmacological studies at Vanderbilt University in Nashville. And we came pretty close together to suggesting the same strategy of using low-dose aspirin, not only because this was sufficient to block the thromboxane biosynthesis, but also because we both had evidence that this would spare the biosynthesis of prostacyclin in the vessel wall. And at that time, this was entirely hypothetical, I mean, the role of prostacyclin. And then the important role of prostacyclin as a mechanism of endothelial thromboresistance became apparent with the COX-IB experience, because prostacyclin biosynthesis is largely the product of COX-2 expressed by endothelial cells in response to laminar shear stress. And when you block prostacyclin biosynthesis with a selective COX-2 inhibitor and you leave thromboxane biosynthesis largely intact, you get into cardiovascular problems. So this clearly showed many years after that our strategy of trying to be pharmacologically selective in wanting to spare extra platelet sites of prostanoid biosynthesis was a correct one. But as it always happens when somebody is recognized, both Garrett and I felt privileged in being singled out of a larger group of people who have contributed to these findings, because the development of low-dose aspirin was initially due to our work of the clinical pharmacology of thromboxane inhibition, but then it is the merit of the clinicians, the clinical cardiologists, who actually did the randomized clinical trials to test the clinical efficacy and safety of low-dose aspirin. And the people I would like to mention before closing are what I call the Oxford trio, a wonderful cardiologist, Peter Slight, Rory Collins, and Richard Pito in Oxford, who designed and coordinated the ISIS-2 trial. And then people like Lars Valentin, Tom Mead, Bo Norvig, who in the late 80s and early 90s did several randomized clinical trials of doses between 75 and 100 milligram and proved the efficacy of aspirin, both for coronary and cerebrovascular prevention.
Right, but they did it because it was proven before that it was better, right? So, I mean, everything has to be analyzed with perspective. So everybody has merit, but who sets the fire sets the fire. So was the ceremony nice? I mean, how was there in Paris? How was
it's a tremendously rewarding ceremony, because the members of the French Academy come dressed with Napoleonic.
Yes, I have never been there, but I've been explained that. So tell us.
And it's really something that gives you the sense of the importance what you have done and the importance of the recognition the French Academy wants to give you. With no rush, with a very historical ritual that has probably been preserved throughout many, many years. And the other feeling is that of sharing it with not just a colleague or a competitor, as we were at the beginning, but with a very close friend, because we have shared about 40 years of friendship in addition to professional life.
Right, right. Okay, well, thank you. I have finished with my questions, but we are not over because Filippo has some more questions for you. And give my best to Angelica, okay? Because I like her very much. She's a very, very nice person.
Thank you, Lina.
Filippo?
Yes, Lina. Well, Carlo, a sort of philosophical question. I'd like to come back to what you said before about the future of aspirin. Now, premise, I fully agree with you. I'm pretty sure that aspirin is here to stay. But regarding the interaction between academia and big pharma, my feeling is that this interaction has always been extremely constructive. I mean, the reason why we are doing so well with cardiovascular disease is because of this interaction, which must be, of course, absolutely transparent, but has been extremely constructive. This is a point raised many times also by Jean Braunwald. And we know that, for instance, Sir Charles Baines comes from industry, and as we said before, was awarded the Nobel Prize. So I think that philosophically, this interaction has been instrumental in getting the success we managed to have in the last 20, 30 years. So when you say that there's some push from industry to promote new antiplatelet drugs, well, this is, of course, something which makes sense. But on the other hand, as academic medicine is ready to defend the scientific truth when for some reason this is put under discussion. So I'm perhaps a bit more optimistic than you on the notion that this interaction between industry and academia, of course, must be all extremely transparent, but has been successful in the past, I think will be successful in the future. And when something inappropriate is proposed by big pharma, I think that academia, as academic doctors, we are very good gatekeepers in order to work always in the interest of our patients. Is the same, you have the same philosophy, the same approach?
Well, as you probably know, I did work in industry for about 5 years between my first time at the Catholic University and when I got the chair at the University of Chieti. And learning from the inside of industry, I learned quite a lot because the culture of clinical trials was really developed to a large extent by people in industry. And so I have great admiration for the people working in industry. And I'm grateful for what I learned during these 5 years. My critical attitude is that sometimes the way in which drug development is driven by industry is not ideal. Otherwise, we would not see this number of failures. Every time we read a new issue of the New England Journal of Medicine, there is at least one failed phase 3 clinical trial. And in fact, the next dialogue will be devoted to one of these failed studies. And I think there is an opportunity for a closer interaction between academic medicine and industry in terms of responsibility. There should be shared responsibility, but also shared responsibility for the expenses related to drug development. In addition, what I think is that a fair amount of independent clinical research funded by public funding is an essential component. And I mean, the development of low-dose aspirin, essentially 100% driven by public funding, is an example that A, it can be done, and B, it may take a longer time frame than drug development driven by pharmaceutical companies. But the advantage is that rather than having 2 trials with the same result, we may have 20 trials, 50 trials. The database for aspirin efficacy and safety is quite remarkable. If you think about it, how many drugs in the cardiovascular field have a similar number of randomized clinical trials? So an integration of independent clinical research and further opening of industry towards the medical scientific community seems to me fundamental to improve what we do in the interest of patients. I hate to see 15,000 patients being treated unnecessarily with a new drug that proves to be ineffective when we have a pretty effective treatment. I hate to think of the cost of this exercise, because it's us, the taxpayers, who will pay this cost that will be downloaded onto the cost of the next successful drug on the market. And so if we could improve this, it would be in the interest of all stakeholders, in premise the patients, but then industry and the medical scientific community. So I'm not at odds with you in terms of philosophy. I admire the pharmaceutical industry for what they do. We would not have this wealth of successful and effective drugs if it was not for the investment of the pharmaceutical companies, but the whole process could be substantially improved.
So to summarize, I think we agree on 2 points. First, the interaction between academia and big pharma has been extremely constructive in the interest of our patients. When the scientific truth is challenged, the academia is a good gatekeeper to preserve the scientific truth. I think we agree on these 2 important statements for our audience. But now, Carlo, you are not only a top scientist, you're also an editor of the European Heart Journal. What about your experience with the weekly Journal Scan and the European Heart Journal? Are you happy or not?
I am very happy, because it's it has been a very stimulating engagement, intellectual engagement for the past 4 years now. And this is due to a fantastic team of young and less young cardiologists who belong to the weekly Journal Scan team that include Massimo Volpe, Giovanna Liuzzo, Leda Galliuto, Rocco Vergallo, and Daniela Pedicino. And we interact every single week, including the Christmas week, including...
It's a weekly journal.
Yes. And we have this email exchange once we have decided that 2 of our group will comment on a particular paper. And then the first draft is sent on Friday night or Saturday morning. We review it, we exchange comments. We get a second version, a third version, then eventually we meet Monday night to discuss any pending issues, and then we finalize it. And so although it has 2 authors, it's the work of 6 different persons, including myself. And then we move on to choose the comment for the next week, as we are doing now.
Well, thank you, Carlo. Thanks to you and the team, Weekly Journal Scan team, as this has been a very successful initiative of the European Heart Journal. And I think it is important for our readers, our colleagues, to have every week this window open on the most important paper published in that particular week in general medicine journals like Lancet, New England, JAMA, and others. I think that this window open on these important journals can be extremely useful for our readers. The last question I ask all our guests, suggestions to improve our dialogues?
I think you're doing a wonderful job. Just keep it up.
Thank you, Carlo. Well, by the way, next dialogue will be on October 31st at 17 CET as usual, but the next time will not be dialogue with the top scientist, but is a dialogue led by the weekly journal Scan, which means discussion on an important paper published in a Journal of General Medicine. Can you anticipate what will be the trial you will discuss next time?
Yes, it will be the Oceanic AF trial, which is a trial of factor 11A inhibition in patients with atrial fibrillation at high risk of stroke, in which about 14,000 patients were randomized to receive either this new inhibitor, asundexian, or a direct oral anticoagulant, apixaban, of proven efficacy and safety in these kind of patients. And unfortunately, halfway through, the trial was stopped prematurely because of lack of efficacy. There were many more strokes and systemic embolism episodes in those treated with the new drug as compared to those treated with apixaban. So it will be an opportunity to ask ourselves initially why phase 3 trials of this size that you presume to be based on very solid phase 2 findings may fail. Then ask the question, is this a problem of this particular molecule, or is it a problem of the underlying pathophysiologic hypothesis that inhibition of factor 11A may protect against hemorrhagic complications of the current anticoagulants and yet be effective in terms of reduction of stroke risk. And we have invited the principal investigator of Oceanic AF, plus 2 discussants, Professor Bianca Rocca and Professor Ugo Tencate, who are experts in this field of research and in this particular field of pharmacology. And I'm sure we will have a very stimulating discussion because there are other molecules with the same molecular target which are in development. And I think it's important to explore whether it's more likely to be this particular molecule. It's those findings that may have not chosen the right dose, or it's a general problem of the underlying concept of inhibiting this particular coagulation factor. So stay tuned.
Sure. Sorry, please tell us.
No, I'm telling the audience, stay tuned because
this was a big blow for the field. Yeah. Okay.
Well, I'm sure that the anatomy of this trial will be really interesting for all of us. I look forward to it. Lina, any closing remark?
Well, just to say that it was a great dialogue, that we have enjoyed a career of success, a career of innovation, and that this innovation was translated into the clinics. And that's very important for any investigator. So I want to congratulate, I have told him many times, so now I say it in public. So my congratulations, it has been a pleasure to meet you. And well, today was perfect as well. So nothing else. We are looking for the next dialogue for the discussion of Oceanic. That will be great as well.
Thank you, Lina.
Thank you, Carlos. Thank you, Lina. It's you in 1 month. Bye-bye.
Thank you, Filippo.